bears an image on it, ventral, front, and back dorsal, of a human figure.
It shows a very faint sort of image of a crucified man. And the crucified man calls us to remember Jesus' death and resurrection.
Scientists expected to debunk it. Instead, the Shroud of Turin did something nobody anticipated. It fought back. The geneticists found DNA that shouldn't exist on a medieval cloth. The physicists found an image no energy source in the ancient world could have produced.
The Shroud is back in the headlines in a major way with a just-released study finding that the cloth does indeed date back to the time of Christ, and with artificial intelligence having something to say about the Shroud as well.
The forensic chemists found blood that recorded a level of trauma science didn't have the tools to measure until the 21st century. Every discipline that came to close the case walked away with more questions than they arrived with. And when the most advanced dating technology available in 2022 was finally applied, it didn't confirm the medieval forgery verdict. It demolished it. This is what the evidence actually shows. The face that was hidden, May 28, 1898, Turin, Italy. A lawyer and amateur photographer named Secondo Pia was granted rare permission by King Umberto I to photograph an ancient linen cloth during a public exhibition. Nobody expected much. It was a documentation job. Slow, technical work under bad lighting with a camera nearly the size of a suitcase. Pia hauled his equipment onto scaffolding inside the cathedral, used magnesium flashes to illuminate the dark interior, and exposed 2 large glass plates. Late that night, alone in his darkroom, lit only by the dim glow of a red safety lamp, Pia lowered the first plate into the developing chemicals. As the image began to form, his hands started shaking. He nearly dropped the glass. What was appearing before his eyes should not have existed. On a photographic negative, light becomes dark, and dark becomes light. What you expect to see is a reversed, distorted shadow. What Pia saw instead was a sharp, high-contrast face. Deep-set eyes, gently closed, a broken nose, a mustache and forked beard, bruising visible along the right cheek. For the first time in history, the figure hidden inside that ancient linen was not a faint smear. It was clearly a man, calm, dignified, marked by what appeared to be extreme suffering. Here is the problem that stopped every researcher who examined it after Pia. Any normal image behaves predictably as a negative. Light turns dark, dark turns light, and the result looks distorted and unnatural. The Shroud broke every rule. Photographed as a negative, it appeared as a true, accurate photograph of a real human face. Think about what that actually means. What the naked eye sees on the cloth is already a negative, which means someone would have had to understand the principle of photographic reversal. 800 years before the camera existed, create a flawless reversed image with no means to see, test, or verify the result.
The image on the Shroud is sometimes very hard to see. It just seems to be almost blurry. That if you're too close to it, you don't see the contrast with the surrounding area, so you don't see the image.
And execute it with precision on a piece of linen. The human eye cannot perceive the world in negative. No artist, no craftsman, no scholar alive at the time possessed the knowledge to even conceive of such a technique, let alone pull it off without a single flaw. That was the first crack in the wall. The Shroud did not behave like a painting. It behaved like a photographic plate capturing one frozen instant in time. And that realization sent a tremor through every scientific discipline that would later touch this cloth. But here is what the photograph actually was: a surface, a threshold, the first layer of something much larger. If what you just heard is pulling you in, subscribe now. Because what comes next, the genetics, the blood chemistry, the physics of the image itself, is where the findings stopped making sense, even to the scientists running the tests. This is the evidence nobody anticipated, and it only gets stranger from here. The DNA that shouldn't exist. In the 21st century, speculation finally gave way to something much harder to challenge: genetics, high-energy physics, spectroscopy, forensic molecular analysis. Scientists stopped treating the Shroud as a sacred object and began examining it the way investigators examine a crime scene: a biological data vault. silently holding 2,000 years of history inside, its fibers. In 2015, Professor Barca assembled a team of geneticists and biologists at an Italian research institution and secured unprecedented access to the cloth. Their mission was not to find the DNA of God. Science has no framework for what that evidence would look like. What they aimed to do was rebuild the life story of the cloth itself, where it had traveled, who had handled it, whose hands had carried it across the centuries. Pay close attention to what comes next, because the methodology is what makes the results impossible to dismiss. Using sterile micro-vacuum devices fitted with ultrafine filters, the team gathered microscopic dust, pollen, and organic fragments from deep between the warp and weft threads, areas where ancient material can remain trapped and preserved for thousands of years. Then they focused specifically on mitochondrial DNA, and for a reason that matters enormously. Unlike nuclear DNA, mitochondrial DNA exists in hundreds of copies per cell. It is inherited strictly through the maternal line. It endures far longer in ancient material. It functions like a biological GPS, a strong, dependable marker of geographic origin, population movement, and human migration across millennia. If any genetic information had survived inside those fibers through 2,000 years, this was the technique that would surface it. For weeks, computers ran continuously, decoding millions of nucleotide sequences and comparing them against global genomic databases representing populations across the entire planet. Then, the final maps appeared on screen. When Barca's team saw the results, the lab went silent. Nobody spoke. What they were staring at contradicted every prediction they had made, every single one. This was not the genetic signature of a single person. It was something far more intricate, and something no one had foreseen. The findings were published in Scientific Reports, one of the most respected peer-reviewed journals in the world. Here is what the team had expected going in. If the Shroud were a medieval forgery produced in France, European DNA should have strongly dominated. If it were an authentic relic from Jerusalem, Middle Eastern DNA should have dominated. Neither happened. Here is the part that changes everything. First hit, the Middle East. Haplogroups linked to the Druze, a close-knit, historically secluded community living in Israel, Jordan, Lebanon, and Syria, whose genetics have barely shifted in millennia, a strong marker of geographic origin pointing directly to the ancient Levant. Second hit, Western Europe. Haplogroups consistent with centuries of European contact, the Poor Clare nuns who restored the cloth, the House of Savoy who possessed it, countless pilgrims who honored it over the centuries. That made sense. You could account for that. But here is where it becomes extraordinary. Third hit, North and East Africa. Haplogroup L3, pointing toward Egypt and Ethiopia. That was surprising. Deeply intriguing. It suggested direct interaction with some of the earliest Christian communities on the African continent. The atmosphere in the lab shifted. Fourth hit, South Asia. Haplogroups M39, M56, and R8. Genetic markers typical of the Indian subcontinent. And then the fifth hit arrived, the one nobody in the room could explain. East Asia. Haplogroups D4 and G2A, commonly linked with China. India, Africa, Europe, the Middle East, all encoded into a single ancient cloth. Let that settle for a moment, and then ask yourself the only question that matters. If a forger didn't place it there, who did? A world written on cloth. Pay very close attention to this, because this is where the forgery theory doesn't just weaken, it collapses. No medieval craftsman working in a French abbey around 1350 could have gathered genetic material from China, India, and East Africa. Globalization simply did not exist at that scale. Long-distance travel happened, but not on a scale large, enough to leave such widespread genetic signatures across one piece of linen. Nor could any person have deliberately planted microscopic traces designed to deceive genetic scientists 600 years in the future. The technology to even detect mitochondrial DNA from ancient fibers wouldn't exist for centuries. There was no blueprint to follow, no forgery to execute. The DNA discovered on the Shroud is not random contamination. It is a shared biological memory. When Barca mapped the data geographically, the mystery grew deeper instead of clearer. The distribution of genetic markers matched with striking precision, along what researchers call the Mandylion route, an ancient path scholars had long dismissed as legend. According to early Byzantine, Syrian, and Arabic records, the shroud was folded so that only the face remained visible, a form called the tetradiplon, and displayed inside a special frame. It did not stay in one location, and it did not begin in Europe. Its journey started in Jerusalem. From Jerusalem, the cloth moved to Edessa in the 2nd century, where it remained hidden within the city walls for centuries. And this is where it becomes remarkable. Edessa stood at one of the most important crossroads of the ancient world, the Great Silk Road. Think about what that actually means for what was found on the cloth. Caravans from China loaded with silk, merchants from India carrying spices, Persian diplomats, Arabian traders, pilgrims from every corner of the known world. And many of them came to honor the image believed to protect Edessa. They stood close, they touched the casing, some touched the cloth itself. And with every encounter, microscopic traces, skin cells, hair fragments, droplets of sweat settled into the fabric layer by layer, century after century. The DNA of the entire ancient world collecting on its surface like invisible dust. The journey continued. From Edessa to Constantinople in 944 AD, the greatest city of the ancient world, where people of every race and culture gathered in its markets, churches, and courts. Then, in 1204, during the Fourth Crusade, the city was violently plundered. The shroud vanished. It reappeared in Athens around 1205, passed through the hands of French knights, and finally surfaced in France around 1353 in the possession of Knight Geoffrey de Charny. And here is where the genetic fingerprint delivers its most decisive blow to the forgery theory. The distribution of DNA on the shroud is not scattered noise. It follows a route. A specific historically documented route stretching across 2,000 years and 3 continents. No forger could have designed that. No accident could have produced it. Dust from sandals, robes, and hands left behind by countless people who stood before this face over many centuries. A biological trail that cannot be fabricated. But the genetics were only one layer of the hidden code. The other half was written in plants. And what those plants revealed pointed straight toward a crime scene. Pollen, blood, and a crown of thorns. Professor Avinoam Danin of the Hebrew University of Jerusalem is one of the world's foremost botanical authorities on the plant life of the ancient Near East. When he received the pollen analysis data from the shroud fibers, he examined the findings in his Jerusalem office surrounded by decades of botanical field records. And when the full results came into focus, he knew immediately that the consequences were enormous. Working independently, Danin and Swiss forensic scientist Max Frey had both analyzed pollen grains trapped deep inside the linen fibers. What they cataloged was not a handful of samples. It was 58 different plant species, identified, cataloged, mapped. 17 were native to Europe, expected, given centuries of contact with the cloth. But the majority came from elsewhere. The Middle East, Turkey, the Anatolian steppe, perfectly tracing the ancient route through Edessa, and Constantinople. Here is the detail that stopped the researchers cold. Scientists identified plants that grow nowhere else on Earth, only within a narrow corridor between Jerusalem and Jericho. One species dominated everything, Gundelia tournifortii, a thorny desert thistle. Its pollen made up nearly half of all samples collected, and it was concentrated heavily around the head and shoulders of the figure on the cloth. Pause and sit with that for a moment. Why would thorn pollen saturate the head and shoulders of a burial cloth? Gundelia tournifortii, with its long needle-like spines, is exactly the plant Roman soldiers could have twisted into a mock crown. It blooms near Jerusalem in early spring, precisely during Passover, precisely during the period of the trial and crucifixion recorded in every historical account. A second botanical signature came from Zygophyllum dumosum, a plant native to an extremely limited region, growing only in the Judean desert and parts of the Sinai Peninsula. Its pollen was also discovered in notable quantities. Think about what that means for any forgery theory. No medieval forger working in France could have collected pollen from plants native exclusively to Israel and distributed it invisibly at a microscopic level across a piece of linen. A forger would have used paint. Pollen cannot be painted. It cannot be sprinkled or brushed with any tool available in the 14th century. It works like a sealed evidence tag from a crime scene, a geographic fingerprint that cannot be fabricated, because no forger could have known it would one day be detected. And then there was the blood. For years, skeptics argued the reddish stains were nothing more than pigments, ochre, cinnabar, or tempera mixed with gelatin. That argument collapsed in 2017. Professor Giulio Fanti of the University of Padua, working alongside physicians from a hospital in Trieste, ran the stains through transmission electron microscopy and Raman spectroscopy in a controlled laboratory environment. When the nanoscale results appeared on screen, Fanti and his team found themselves looking at something no pigment hypothesis could survive. Not paint, not ochre, not any fabricated substance. Human blood. Type AB. One of the rarest blood groups in the world. This same blood type appears repeatedly on ancient Christian relics, including the Sudarium of Oviedo, a cloth traditionally believed to have covered Christ's face at burial. But what truly stunned the team was not the blood type itself. It was what the blood contained. Pay close attention to what comes next. Scientists discovered nanoparticles of creatinine and ferritin bound to hemoglobin. Such extreme concentrations occur under only one set of conditions: catastrophic, fatal trauma, prolonged torture combined with severe dehydration and massive muscle destruction. When muscle tissue is repeatedly damaged in a process called rhabdomyolysis, creatinine floods the bloodstream in enormous amounts. The blood preserved on the shroud recorded exactly that physiological cascade. This is not symbolism. This is not iconography. This is a biochemical signature of agony. The man wrapped in this cloth had been beaten to a physical condition already incompatible with life before crucifixion even began. Evidence consistent with more than 100 blows delivered by the flagrum, Roman leather whips embedded with lead weights, and the body recorded every impact at the molecular level. And then there is the detail that still troubles researchers. The blood stayed red, which, for ancient blood, should be impossible. Normally, old blood darkens to brown or black within weeks, but scientific analysis revealed unusually high levels of bilirubin, a substance released by the liver during extreme stress and catastrophic physical trauma. Bilirubin preserves the red color of blood. For centuries, the blood of a tortured man stays red. Not mysticism, biochemistry under unbearable stress. A painter can imitate the appearance of a wound. No human hand can recreate the biochemical signature of extreme trauma, organ failure, and hypovolemic shock. Every molecule in that blood pointed to the same place, and the same event. But there was still one test that the world believed had settled the question permanently. One test that told everyone to move on. It got the answer completely wrong. The test that got it wrong. In 1988, three of the world's most prestigious scientific institutions, Oxford University, the University of Zurich, and the University of Arizona, conducted carbon-14 dating on a fabric sample from the shroud. Their conclusion was definitive. The cloth dated to between 1260 and 1390 AD. Medieval, a forgery, case closed, the world accepted it. The Catholic Church stepped back, referring to the shroud as an icon rather than a confirmed relic. Skeptics celebrated, believers retreated, but science never stands still. 3 decades later, researchers identified exactly where the error had occurred. And here is the critical detail. The problem was not the technology. The carbon-14 process itself was sound. The problem was where the sample had been cut. In 1988, a piece of fabric no larger than a postage stamp was removed from the very edge of the shroud, a corner that had been handled countless times over the centuries, absorbing sweat, skin oils, candle wax, and bacteria from hundreds of years of human contact. Then Raymond Rogers, a chemist at Los Alamos National Laboratory in New Mexico, examined that corner under his microscope, and what he found was devastating. That corner had been carefully repaired during the Middle Ages. The Poor Clare nuns, working to preserve the damaged edge, had woven new cotton threads directly into the original fabric, dyed with alizarin, and sealed with gum arabic to match the aged linen in color and texture. The repair was meticulous, invisible to the naked eye. Here is the point that cannot be overstated. The main body of the shroud contains no cotton at all. It is made entirely of linen. The 3 laboratories didn't date the shroud. they dated the patch. The entire 1988 verdict, the medieval forgery conclusion that shaped public understanding of the Shroud for a generation, was built on a contaminated sample from a medieval repair job. And then, in 2022, the ground shifted again. Physicist Liberato De Caro at the Institute of Crystallography in Bari, Italy, introduced a completely different analytical approach: wide-angle X-ray scattering. Instead of measuring radioactive decay or examining surface contaminants, this method studies the aging of the linen cellulose itself at the atomic level. Over time, linen naturally deteriorates. The polymer chains of cellulose break apart slowly under exposure to radiation, humidity, and temperature. This process functions as the material's internal clock, entirely separate from any contamination or repair work on the surface. De Caro compared the Shroud samples against fabrics of precisely known age, from Egyptian mummy wrappings dated to 3000 BC through to verified medieval textiles from the 13th and 14th centuries. When the aging curve appeared on his monitor at the Institute of Crystallography in Bari, he called his colleagues over immediately. The Shroud's molecular structure did not match medieval fabrics, not even close. It was dramatically older. It matched linen recovered from the fortress of Masada in Israel, dated between 55 and 74 AD, the 1st century, the time of Christ. In 1988, science declared the question answered. In 2022, it reopened it with a method the 1988 teams never had. But one mystery still stands above all the others, and this is the one modern physics genuinely cannot explain: the impossible image. No brush strokes, no pigments, no ink, no dye, no transfer medium of any kind. The image on the Shroud of Turin exists only on the outermost 200 nanometers of the linen fibers. To give you a reference point, a single human hair is approximately 70,000 nanometers wide. The image occupies a layer hundreds of times thinner than that. It is not painted onto the cloth. It is a chemical transformation at the molecular surface, caused by oxidation and dehydration, a kind of scorch signature left behind by an unknown source of energy. Scientists at Italy's National Agency for New Technologies, Energy, and Sustainable Economic Development devoted years to recreating it. They tried acids, extreme heat, gamma radiation, ultraviolet light from every standard source available. Nothing worked. Nothing came close to producing an image with the same properties. The only approach that generated even a partial resemblance was a brief, intense pulse of vacuum ultraviolet radiation. Technology that did not exist in the ancient world. Technology that barely exists in controlled laboratory conditions today. And here's what makes the image even more extraordinary: to imprint across nearly 4 square meters of fabric with the precision that the Shroud displays, the energy source would have had to release its burst in less than 1 billionth of a second, powerful enough to mark every fiber at the surface level, precise enough not to burn a single thread beneath. In 1976, NASA scientists applied image analysis technology developed for space photography to the Shroud. What they discovered changed the terms of the debate permanently.
This image zooms off the view screen in this amazing 3D contour. No painting has it, no artwork has it. It is totally unique to the Shroud.
The image encodes perfectly accurate 3-dimensional information. The intensity of the imprint at every point on the cloth corresponds exactly to the distance between the body and the fabric at that point. Not approximately, exactly. The spatial data embedded in a 2-dimensional piece of linen is accurate enough to reconstruct a full 3-dimensional model of the figure beneath. No artist, ancient or modern, has ever produced anything like this. Every attempt to recreate it in a laboratory environment has failed. Think about what that actually means. Digital analysis of the image also identified shapes over the eye region consistent with coins. Specifically, lepton coins minted under Pontius Pilate around 29 AD. These coins circulated in a specific region during a brief window of time before being pulled from use. The chance that a medieval forger could have known this numismatic detail, let alone encoded it invisibly into a linen cloth at a scale detectable only by 20th century imaging technology, is not small. It is effectively zero. The anatomical evidence tells the same story. The nail wounds on the Shroud passed through the wrists, not the palms. Forensic science now confirms that nails through the palms cannot support body weight during crucifixion. Every single medieval painting of the crucifixion places the nails through the palms, because that is what medieval artists believed based on the iconographic tradition they inherited. The shroud shows the anatomically correct location. Even the retraction of the thumbs is visible, caused by nail damage to the median nerve. This is a physiological response that no medieval artist could have understood, because the anatomical science required to understand it would not be developed for another 600 years. Layer by layer, biology, chemistry, physics, botany, numismatics, forensic anatomy, everything converges on one city and one narrow window of time: Jerusalem, between 30 and 33 AD. What no one can easily dismiss. Here is everything a medieval forger working in France would have needed to pull this off. Create a photographic negative 8 centuries before photography existed. Embed pollen from plant species that grow exclusively between Jerusalem and Jericho. Distributed at a microscopic level no medieval tool could achieve. Produce blood containing trauma biomarkers, creatinine and ferritin concentrations consistent with rhabdomyolysis that would not be discovered by science for another 600 years. Encode 3-dimensional spatial information into the surface of a cloth with a precision that no modern laboratory has been able to duplicate. Leave coins of Pontius Pilate over the eye region at a scale visible only to 20th century digital imaging. Show anatomically correct nail placement through the wrists instead of the palms, despite every medieval artistic tradition placing them in the palms. Preserve the biochemical evidence of rhabdomyolysis, hypovolemic shock, and bilirubin-stained blood through 20 centuries of storage, and somehow gather DNA from China, India, East Africa, the Indian subcontinent, and the Middle East, all from a French abbey, with a candle and a paintbrush. The Shroud of Turin is not a painting. It is a forensic record of a real human being, a biological archive, a silent witness to a single moment that has shaped more of human history than any other event in recorded time. The DNA mapped across its fibers traces, a 2,000-year journey through the crossroads of the ancient world. The pollen places it in Jerusalem during Passover. The blood records the biochemistry of someone tortured beyond survival before crucifixion began. The image cannot be explained by any energy source available in the ancient world, or, as it turns out, in the modern one. A painter can imitate the appearance of a wound. No human hand can recreate the biochemical signature of extreme trauma, organ failure, and hypovolemic shock. So, this is the question the evidence leaves behind. Do you believe science will one day produce a natural explanation for what happened inside that tomb? Or are there moments in history that lie permanently beyond the reach of any instrument we will ever build? Leave your answer in the comments. And if this video raised more questions than it answered, the next one goes somewhere even the researchers weren't prepared for.